All right, where are we at today? Dum, dum, dum. Oh, sorry, yawning already. Jeez, haven't even started yet. Let's put that scary image up for a little while. Come on, where's my chat gone? I don't know why. Oh, there it is. Oh, yeah, I have coffee. Ta-da! Just haven't had time to drink it yet. Yeah, all right, all right. Cat is demanding attention. There we go. Watched a good film last night with Jake Gyllenhaal called End of Watch on Netflix. So that was pretty cool. All right, ladies and gents. No time like the present, as they say. No rest for the wicked. Okay, so essentially what we're going to do today is begin the last and final section of the course. All right, we do have an exam today, so there's only going to be one lecture this morning. Lecture, you know what I mean. And we'll basically kind of get started on the material, which will then continue going through next week. Right, so none of this material is going to be on the exam today. That's all on stuff just up until the end of the last lecture. Oh, sorry, cat issues. Okay, so essentially what this section is all about is really putting together the kind of all the bits and pieces that we've learned, right? So really, we spent a lot of time looking at, kind of like the molecular genetics, the classical genetics, kind of mutations, alleles, phenotypes. What else? Kind of how DNA is replicated, inherent errors in that, the mechanics of transcription and translation, right? And so really that's kind of like all the nuts and bolts, but we haven't really turned it into a picture yet. Right? And so that's really what this section is all about. And large part, what we're going to be looking at is control of gene expression. And then we'll look at some kind of bigger scale stuff, some genomics and things like that later on, which is fun to look at. So the reason why gene expression is kind of the bigger picture is that basically you've got all this DNA, right? You've got a collection of genes, you have a collection of different alleles, right? For those genes, which are, shared and different between you and other individuals, depending on your relatedness. But you are all kind of more or less the same, right? You are very similar in terms of body structure, body plan, what you look like. You respond in very similar ways to different stresses, different kind of environmental insults, hunger, heat stress, et cetera, right? But yeah, you are also different, right? And so how do we go from that kind of pictures of nuts and bolts of, you got this and you got this and you got this, and this does that and that does this, to kind of, who you are kind of as an individual. And we use ourselves as examples because it's kind of like an easy context, right? But you can apply this to anything. You can apply it to baker's yeast, right? Or even brewer's yeast. That's a fun one because You know, some of you have like drinking Miller Lite or something, which is fair enough. You know, a nice hot day. What's that? 3.5%, 4% maybe. Others amongst you maybe scorn Miller Lite, right? And they might like a nice, you know, chunky triple double mega dollop IPA, which is like up to 8% or something crazy, right? Gets you drunk in an instant, right? The reason being is that those yeast used to make those different beers, although they are, you know, they're all what are they? Saccharomyces pombe. I can't remember what brewer's yeast is. They are different, right? So they are the same species. Shoop IPA. Yeah, I'm not a big IPA fan. I lived in Pacific Northwest, and yeah, they went a little bit nuts on the whole bitter IPA thing. But so the flavors come from the hops, but the alcohol percentage comes from the yeast, and also, you know, how they're brewed to some extent as well. But essentially, any organism you look at, you'll have variations of form and function, right? And so what causes that, right? Why am I, you know, over 6 foot tall and, you know, other people around are like 5 foot tall, right? We have the same genes. You know, we are 99.99% identical, perhaps even more so, right? And yet somehow I'm over a foot taller than that individual, right? And so this is really, actually, it's a very scary, kind of complicated, like intimidating place to wander around in terms of biology. But this is where the control of gene expression comes in, right? Because even though, you know, I and my wife, you know, who's quite a lot shorter than I am, have the same genes for, I don't know, bone growth, body plan, you know, all of that jazz. I'm a lot taller than her. So I must have some variation of that gene. Maybe it works better. Maybe we're talking about changes in protein structure and function. It's possible. But most likely it's different in how it's expressed, right? That may be because the protein, is the gene itself resulting in, you know, kind of bone growth is expressed more highly. It may be because a regulator that stops that from being expressed is not as effective or is expressed at a lower level, or a whole bunch of things, right? And so essentially what I want to get across as big picture stuff, because we're going to be going way into the weeds, like some of this stuff is like like kind of like tiny stuff, right? But I want you to hold in mind this idea that essentially you are you as a complex organism as a result of thousands of rounds of cell division from one cell, your original fertilized zygote in your mother, right, to the organism that you are now, right? And those cells have all kinds of different uh, identities. They have different gene expression profiles. They all have the same DNA, right? More or less, apart from red blood cells. Those don't have any nuclear DNA, at least. But they're weird. We don't talk about red blood cells much. They have like they have the same DNA, but they are obviously very, very different, right? So how does that happen? How do you get legs where you have legs but not arms? They're both limbs. Why do you have them on, you know, both sides? Why are we bilaterally symmetrical? Why do you have, you know, a shoulder here and not like, you know, around where your ribs are, right? It's all about what genes are turned on. Yay, Hox genes, definitely. Body patterning. Actually, that's a good, it's a cool example because that's why snakes don't have legs, right? Essentially, the Hox gene, which specifies kind of the shoulder and pelvic girdles, is doesn't work very well, or doesn't work at all. It's suppressed by other genes. That's how snakes lost their legs, essentially. Maybe it's how whales sort of lost their legs, you know, even though they kind of have legs as flippers, but kind of don't as well. You know, so it's really, it's interesting from just a basic, wow, how does this work kind of approach, why does this turn into this and not that? It's really interesting from an evolutionary perspective as well, because we are the accumulation of tens, hundreds, thousands, millions maybe of differences from our series of last common ancestors. And some of those differences are what make us who we are now. So we can kind of look at this from a evolutionary perspective too, right? And so it's just very, very interesting. Even again, all right, I'm kind of going off on this kind of comes more from developmental biology. Even the number of vertebrae you have, I can't remember, how many do humans have? I'm not much of an A&P person, like 30, something like that, 27. I can't remember exactly. A bunch. You look at a snake skeleton, it's got crazy numbers of vertebrae, right? It's got ribs all over the place, right? More ribs than you could count almost. And so it's not the genes which control vertebrate development or formation that change between snakes and us. It's the clock, right? So the reason why snakes have more vertebrae than we do is that their clock that specifies, where there will be a vertebra, and it's kind of done in chunks, runs faster than ours does. That's pretty much it. It goes tick, 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 tick. Ours goes tick, tick, tick. And mice goes tick, 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 tick. So it's not the genes itself that control the development. It's the genes that control the control of the development, if you see what I mean. Right. And so it's a hugely complex and kind of scary field to kind of dip your toes into, to be honest, right? There's an awful lot going on, and the more you know, the more you realize what you don't know. So we're not going to go too far into that aspect of things in this section, simply because we don't have time. I mean, I in Dev Bio I spend like at least 5 lectures on Evo Devo, 4 lectures maybe, and that's after an entire semester of talking about control of development. But I just want you to kind of keep in mind that kind of big picture context, right? Gene regulation or control of gene expression is how we are, who we are, and how we respond to our environment, right? At a kind of cellular and organismal level, right? You can look at like the whole, and you'll see a particular phenotype, right? Height maybe, or skin color, complexion, who knows what, any kind of things, right? But those big picture stuffs are the result of differences in gene regulation between individuals and between species, between genera, and so on and so forth, right? So kind of keep that in mind, and hopefully some of this will be a little bit less intimidating. But there's a lot of detail here, and some of these concepts are tricky. So in particular, we're going to go into quite some detail of gene reg of prokaryotes, because there's a couple of really good systems here which will teach you some of the kind of the basic concepts, I think, of control of transcription, right? And the reason why we do those is because they're relatively simple, even though that may not seem so to you at the time, right? Because as you can see here, there's a ton of stuff going on, right? But the reason why we do this first is because the principles are relatively straightforward. As soon as we get into eukaryotes, it's just like batshit crazy, basically. In fact, funny story that I typically tell students is that when I first started at Penn State as a postdoc, there were emails going around about journal clubs, which are, basically you get together and each person presents a paper every week. And so you kind of get an in-depth view on the research going on in the field. And there was an email went around for the gene reg or gene regulation journal club. I was like, oh, that sounds really cool. I talked to this grad student. She's like, hell no, stay away from that. It's crazy. Those people are nuts. You don't have anything to do with them whatsoever. I was like, oh, okay. And I didn't. And there was like 5 or 6, large research labs all working on that aspect of biology, and they really truly went into the weeds, kind of way beyond the level which I, would, probably be able to cope. Anyway, that was, sorry, I got. Probably he's got stuff with cat fur or something, isn't they, Fuzzbun? Okay, so essentially, I mean, this is, don't get too scared by this. We'll come back to it. It's a nice picture, and we'll look at it in a little bit. But essentially, what I've been talking about really is, what's the purpose of gene regulation, right? Where are these genes going to get expressed? Not all genes are expressed in all cells, particularly when you talk about a multicellular organism, right? Genes resulting in detoxification of, I don't know, alcohol, let's say, are expressed in the liver. They're not expressed in the skin, right? And so, what genes are going to be expressed? What are the levels of control along that process? Where is that protein going to be? How much of this do we need, right? When is it going to be turned on, right? Some things are conditional, some things are not. Those are often called they're called housekeeping genes, not because they go around and like tidy the place up, but because they're always being expressed, because they're always needed. Things like ribosomal proteins, good examples for that, right? And there's some of this going on in prokaryotes. A lot of this kind of really more applies to eukaryotes, but there's a lot of different steps at which control of gene expression occurs, right? Typically, in prokaryotes, we're going to be talking a lot about this aspect, right? Transcriptional regulation, which is the kind of conventional way of thinking about gene regulation. When or where do you turn this gene on, right? And that's a very important one too. It's not to be trivialized, but it's only part of the picture, because you've got all kinds of other stuff going on. You've got RNA processing and splicing, right? So we're going to talk a little bit about alternative splice isoforms later next week. Hey, leave it alone. For example, just bored. Where you could talk about how translation is controlled or not, how long that mRNA transcript hangs around for, because obviously that's going to affect how much protein is produced from it. And even when all of that is done, there are things which need to be done to certain proteins for them to be functional. And even once they are functional, those proteins can be turned on and off, right? And so there's just, it's just absolutely crazy how much stuff is going on, which kind of makes sense, right? Because we are complex, highly differentiated organisms with, I don't know, hundreds of cell types. I'm actually, again, I'm not much of an AMP person. But you just got to look at just what you can see on the outside, and there's a whole bunch of different stuff going on, right? And so that's kind of part of the way in which we can explain that. Okay, so we're going to talk about some general concepts first, right? And if anybody has any questions, do please, again, stop me before I kind of continue rambling along too far, so I can go over something. Oh, coffee's sinking in. Hang on. So anyway, essentially we've got different ways of thinking about gene regulation, right? We can consider whether that gene is on or off. I don't know why my ears kind of keeps blocking up. We could think about, hang on, going to keep losing my train of thought. So is the gene off? Then you need to positively regulate it to turn it on, right? So you need something to activate it. God, I don't know what's going on with my ear right now. It's like I've got feedback going on. There are other genes, and this is typically more common in prokaryotes, which are always on, right? And so they need to be turned off. Otherwise, you might produce them when you don't need to. I'm sorry, this is really difficult because I kind of weird. Hang on. You don't want to see me do that. Hang on, look at my cat instead. Kitty cam. Okay, Noel, you're in charge. So, how did she do? Should she explain everything all right? Oh yeah, that's one of my favorite Marvel films. Let's get things back in control. Hopefully that's gone away because that was really annoying. All right, so essentially what you need to think about is that gene normally on or off? If it's off, which is most eukaryotes, they need to turn it on, right? So you need some kind of protein to help you do that. If it's always on, then you need to have some way of turning it on. So now you're going to need a repressor, right? And so there's a bunch of different words that we're going to be using a lot in this to describe what's going on. So this is part of kind of like the lingo, essentially. So there's a nice little graphic, right? Essentially, if you have positive regulation, so you're regulating it in a positive way, so you're making expression happen. Without that protein being present, you don't have any expression, right? So this allows you to turn genes on only when they're needed, right? The other way round of thinking about it is that maybe you need, and this is more typical in prokaryotes, maybe you always need something, right? Maybe some metabolite. We're going to talk about tryptophan, amino acid in a little bit. And but sometimes, you're gonna outstrip your demand or your need, and so you need to turn it off. Otherwise, you just produce stuff that you don't need, right? And so now you're going to negatively regulate that. We also have, here's another couple of words, inducible and repressible, right? So in positive regulation, right, we're talking about there's nothing there, so we have no transcription, right? And so we need to activate it with a transcriptional activator protein. Now, it could also be off because there's something actually bound there which, say, prevents the binding of this protein, right? So it physically occludes that bit of DNA that activator protein will recognize, right? And that's pretty common, particularly in eukaryotes. So in that case, now you need to induce that repressor to leave, or essentially, yeah, you need to make that repressor leave. Now, you can have the opposite, right? In which case that gene is always on, and now you need to repress it. So now you have what's called an aporepressor, right? So aporepressors, and this is an important one because we'll talk about this. I think that's the lac operon. I have to kind of refresh my memory in a little bit. It's easy to get confused between the two. Essentially, you have a protein which doesn't bind its regulatory sequence on its own, right? That's why it's not called a repressor, but an aporepressor, right? And so this is, I'll type that out just so we have a copy of it. So essentially it's present in the cell, but it doesn't do anything because its co-repressor is absent, right? And so only when its co-repressor, which may be the end product of that metabolic pathway or process, only when that is present does it then turn off or repress transcription, right? So keep those in mind. They're slightly tricky concepts to get because you're thinking about, where we talk about positive or negative regulation, we're thinking about what's the default state of transcription, right? Does it need something to turn on, or is it always on and needs something to turn off? When we're talking about inducible versus repressible, now we're talking about what thing is coming in to control that gene expression, right? Whether it's an inducer or an aporepressor with co-repressor. So here's a kind of good example of that, right? So remember here I was saying that positive regulation stuff needs to be turned on. Well, in inducible transcription, sorry, I got whatever's going on with my ear back again. I think it's all the tree pollen around, kind of allergies or something, I guess. So anyway, that repressor is bound in the default state, right? So transcription is off. Now you need an inducer to come in to remove that repressor, right? So you induce transcription to occur. And typically that repressor is actually physically blocking. That's actually one of the main ways in which repressors work, is that they either block regulatory sequences, so they prevent activation of transcription, or they block the RNA polymerase binding site or other aspects of where the machinery kind of assembles. So that's inducible. In repressible, essentially here you have transcription occurring normally, right? So this is the kind of the opposite. And that aporepressor is unable to bind, right? It's not in the correct transformational state. Sorry, conformational state, not transformational. And so when that co-repressor is present, and again, that could be a metabolite, it doesn't have to be a protein or anything else. It could be an ion even, right? It could be something as simple as that. When that is present, it binds to the aporepressor, and essentially it changes its shape a little bit. So now it's able to recognize its bit of DNA to bind to, and it binds to the regulatory region and stops transcription. Does anybody have any questions about any of that? I'm just going to have to get used to my ear being loud. Oh, now's a nice coffee. No, okay. There's also different kinds of feedback loops that can occur as well. So a good way of thinking about this is basically controlling. You can have positive feedback or you can have negative feedback, right? So let's say you've turned on a gene and it's involved in producing, I don't know, amino acid, right? But once you have enough of that amino acid, right, you're essentially fulfilling the need of the cell in terms of translation. Any extra amino acid is just going to be wasted, right? It takes energy to make and resources and things like that. So you want to control how much of it is there. And so that amino acid can then act as part of the regulatory complex, which then shuts it down, right? So it's that amino acid or product can be the co-repressor that we're talking about here, right? And so essentially when you get enough transcription, this builds up to a high enough level to bind the apo-repressor and shut down transcription, right? That's quite common in prokaryotes as well. You could also have a positive feedback loop, right? So imagine that you want to produce something very, very quickly. I don't know, like say an immune response. Then you want to kind of increase the amount of something you produce in response to increasing the amount of what you produce. Those are much rarer typically because they're much more context specific, right? Typically you want something very quickly, and that would be one way of doing so. Okay, let's just have a think. And again, just to reiterate, typically negative regulation is more common in prokaryotes. So most genes in prokaryotes are on until they're told to be turned off. The opposite is the case in eukaryotes. Most genes in eukaryotes are off until they're told to be on, right? So they're positively regulated. And part of that is, I guess you could think about in terms of the complexity of those organisms. You can get away with negative regulation more easily when you have, you know, a few thousand genes maybe, and you're able to coordinate gene expression within a cell, a single cell, essentially. For eukaryotes, which are considerably more complex, that just wouldn't work, right? It'd be really, really hard to make sure you're turning off all the genes that you need to turn off to get what you need to be turned on, if you see what I mean. It's a lot easier conceptually to just turn on the ones that you need. When you have, like humans do about 20,000 genes or so. Okay, so again, just to recap a little bit in prokaryotes, most, not all, but most gene activity is controlled through control of transcription. And quite often, actually, there are, and what we'll see where we go through the lac operon and the trip operon, is there are usually two main methods of controlling gene expression in, or for each operon, there are typically two ways of controlling the expression of that operon. You'll have a big on-off switch, essentially, right? Gene is on, turn off, bang, right? That's a very crude, like a large fold change in transcription type switch. Very much like an on-off switch, like the big kind of blade switches they have in Frankenstein movies and stuff like that. There'll also almost always be a second finer tune control of gene expression, right? So there you don't want it to be completely on, you don't want it to be completely off, you want it somewhere kind of in the middle, like just off and on again. Sorry, that's a Creature Comforts. Actually, you should check this out. I'll put it in here because it's fun. See if I can quickly find one just because it's pretty awesome. What was it? It was a jaguar. Is it jaguar? Ha ha ha. Puma. There you go. Sorry, I've got to show this to you just ever so quickly. Where we at? It's got absolutely nothing to do with anything that we're studying. It's just funny. Sorry, I just love watching that. Always makes me laugh. So this is by, oh, here you go. You can watch. I don't know why that doesn't work so well in Zoom. The sound of what I'm watching doesn't come through. But yeah, watch that later. It's basically Creature Comforts is from Aardman Animation, which is the studio that does Wallace and Gromit, based in Bristol in the UK. And essentially, they wandered around the UK, London, actually all over the country, and they interviewed people, like kind of on the street. I just asked them questions about stuff, and then they put that recording to an animal, right? So there's a bouncer for a nightclub, for example, gets lip synced to a hamster, right? It's in this like little leather jacket. It's like really like full on kind of scary little hamster. And that one was as far as I can remember, a Brazilian exchange student who had come over from Brazil and was basically complaining about how crap the weather was and how terrible the food was. So anyway, check those out when you have time. They're always kind of fun things to look at. Part of your cultural education, right? It's important. Got to educate the whole of you. Anyway, bit of a tangent there. So yeah, that's where the off and on again comes on, because it was basically some turtles talking about how they like their temperature just right in their enclosure, because you go off and on again. Anyway, so there's kind of those typically two means for controlling gene expression in any given operon. Not always, but a lot of the cases, you've got like a big brush stroke, big kind of large fold change control, which is typically in the 100 to 1,000 fold change in gene expression, right? So it's a very, very large change. And then you have the finer control, which typically nets you like say a 10-fold change in gene expression, right? So it's kind of really that getting the right level, because it matters a lot, right? Because this is expensive, producing proteins. And particularly in bacteria, right? If they are producing proteins that they don't need, then they'll lose out in terms of competitiveness with other bacteria around them, right? So they'll essentially get crowded out from their whatever environmental niche they're in. Okay, so to remind you again, bacterial mRNA is polycistronic, right? So all of the genes typically in a metabolic pathway will be organized into an operon, right? And from that operon, you will get one single mRNA transcript, right? Which will contain the cistrons or the coding sequences for each of those genes. So really it's a kind of it's a very neat way of controlling expression of all things that you need to be expressed for a given process or task. And that's called coordinate regulation. It happens in eukaryotes too, but the mechanism in eukaryotes is very different because those genes are scattered around and instead in eukaryotes, it's controlled by the production of transcription factors or activity of, right? In bacteria is controlled by turning on or off or controlling, fine tuning the whole operon at once, right? So you got one regulatory sequence, and then you've got all of the genes essentially that are required to produce proteins to do whatever job it is. So again, that's a very big difference between eukaryotes and prokaryotes. Oops, get you back. Okay, so this is basically the picture that we started with, kind of way up at the beginning of the PowerPoint. This is a very classical layout for a prokaryotic gene regulation system. So you will have an operon over here, right, which is all of your structural genes, whatever they may be, whatever they may encode. You have an operator, right? So this is kind of your regulatory region, would be the best way of describing it. You have your operator, which is typically where repressors or inducers bind. And then you have a promoter, which is where your RNA polymerase binds, right? That's where the your TADA box is, essentially. You also, over here, and this doesn't actually have to be right next to the gene, sometimes, the operon, sorry. Sometimes it is, other times it can be elsewhere in the bacterial genome. It doesn't actually matter. And this is another gene, right? And so this is a gene that codes for a protein, right? Has its own promoter or regulatory region, and it produces the regulatory protein, I guess, which could be an inducer or a repressor. And that typically binds to the operator, right? So you have this two-part system. You have the operon, which encodes the stuff that needs to be made, right? And has the regulatory regions to control the expression of those genes. And you also have a separate regulator gene, right? Which produces a protein which is involved in the regulation of that operon, right? And then down here, you obviously can see you get your polycistronic mRNA that gets translated into distinct proteins, and those catalyze different steps of a pathway, let's say. And quite often, this product Y, right, the end product of that process, is involved in the feedback loop or the control of regulation of that operon. Not always, but most of the time. Okay, so we're going to look at two different systems yet, right? We have a, hang on, let me just get the picture. Here we go. All right, so this is what the lac operon looks like. And this is a good example of a inducible system, right? So this is basically on, but it's repressed by this repressor protein. And then we require lactose coming in, which is our inducer for transcription to occur, right? So whereas the trip operon is the opposite. But so this is a, both of these examples are really good ones. They're very well studied, well, mostly. There's actually one gene in here called, was it LAC X? Anyway, one of them, which we actually don't know what it does, which is kind of funny. But essentially, there's our operon, which has 3 genes in it, one of which is a lactose transporter protein, right? So this is called lactose permease, right? So it allows lactose to get into the cell. And then we've got a enzyme which breaks lactose apart into galactose and glucose, right? And so that's beta galactosidase, right? So, and there's another one as well. We don't actually know what that one does, right? So we've got 3 structural genes, the permease, the galactosidase, and then something else, right? And so that's what LAC-Z, LAC-Y, and I think LAC-X, can't remember what it's called. And we also have a bunch of regulatory elements, right? We have our repressor gene called LAC-I, right? So the LAC-I, this would be LAC-I in the LAC operon, right? This separate regulator gene. And we also have, a promoter and an operator, which is LAC-O. So this would be LAC-O here, and LAC-P, which is the LAC promoter here. Right, give you a few moments to kind of take that in. Again, any questions, just shout them out. All right, and this is just a description of what I just told you with regard to this. Right, so essentially what we need as if we were a bacterium, glucose is the best. Oh, hello, Jasmine. Where she is hiding. It's a lot easier to use. This is what we want to end up with, essentially, right? Which is the most basic form of carbohydrate energy, right? So if the bacterium has glucose to survive on, then it will preferentially use glucose because there's a lot fewer steps involved in the metabolism of glucose. However, and so if lactose isn't present and glucose is, then we don't need any of this jazz, right? Even if lactose is present, but we also have glucose, then we don't need as much of this, right? Because we have glucose to use. So essentially what the bacterium needs to do is go, okay, well, what do I have around me, right? Do I have glucose? If so, I don't need any of this lactose shit, right? I'm going to use glucose. It's way better, right? Don't have glucose, well, that sucks, right? Because glucose is good. Don't have lactose either, right? So it's still, I don't need any of this lactose stuff, right? This beta-galactosidase, permease, things like that. What about if I don't have glucose, but I do have lactose? Okay, now we need to turn this on, and we need to turn it on a lot, right? Because we need some form of energy source to survive. So now we're going to be essentially turning on this whole operon to make a lot of this product so that we could start A, importing lactose into the cell, and B, breaking it down into glucose so we can use that glucose to produce ATP via cellular respiration. Now, at some point, we're going to do such a good job of that that glucose levels are going to start rising. And that's essentially telling us that, oh, okay, we can kind of dial back on the lac operon stuff a little bit, right? Because we now have more glucose present. Or maybe the environment has changed and glucose is now present at a higher concentration, right? So now we're going to feed back onto the lac operon and just kind of turn things down a little bit, right? So hopefully now you can see that kind of decision-making process that occurs at a cellular level. Do we have A? If so, we don't need to do what is involved in making B, right, or using B. If we don't have A, is B present, right? Because if it isn't, then there's no point producing all that stuff. Only if we don't have A and have B present do we turn this on, right? And so there are, again, two main control mechanisms for regulating the lac operon. One is presence of lactose, right? That's the big one, right? That's what turns this thing on gangbusters. The second one is presence of glucose, and that's more the kind of fine control deal, right? So keep that in mind. Remember the two different ways of controlling expression from the lac operon. Okay. And again, please ask questions, right? This is, again, like a lot of the stuff that we've studied, you know, if you kind of go through it step by step, it's not too bad. If you look at it as a whole, it's like, holy crap, you know, it's really overwhelming. And it's hard to figure out exactly where everything is. So I'm trying to keep your focused on the context, like the bigger picture of it, so you don't get too overwhelmed with the details. So essentially, the LAC-I protein, right, which is shown here, actually attached, like contiguous with the LAC operon, it will bind the operator and stop transcription, right? So this occurs in the absence of lactose, right? So if there's no lactose present, you don't want any expression from the LAC operon because it would be a waste of time, waste of energy, more specifically, right? You'll be producing something that you can't use because there's no lactose present, right? So the LAC-I protein or LAC-I repressor is bound to the operator in the absence, oh, it's A, LAC-A, in the absence of lactose. Now, when you start detecting lactose in the environment, right, there is some around, now that lactose is going to bind to this repressor, right, and result in it falling off of the promoter. It basically changes its shape. Similar to how repressible systems, the apo repressor changes shape in the presence of the co-repressor. The repressor in the inducible system, sorry, changes shape in the presence of the inducer, no longer recognizes the DNA. So it will fall off, and now RNA polymerase is able to assemble, and you'll get transcription, right? So this is the big switch. I can't remember what the fold change is. Don't have it down there. It's in the several hundreds of fold change and transcription as a result of the presence of lactose, right? So this is a big on-off switch. Okay, now we have something a little bit tricky to kind of get at. Okay, so we're talking about the big on-off switch here, but as a result of the expression of the LAC operon, right, we're going to be importing and digesting lactose into something we don't need, galactose, I'm not sure what that does, and glucose, right? And so at some point, we're going to do, if we were a bacterium, we're going to do such a good job of that glucose levels will start rising. And so now we need to start dialing things back a little bit, right? And so we need to detect glucose levels. The problem is glucose is actually kind of hard to detect on its own. It's a carbohydrate. It's not particularly interesting or having a unique shape. So there's no real easy way that we can detect it as itself. However, what we can detect is the consequence of having more glucose. And so this is something that's also shared with eukaryotes. There's actually a kind of like cellular energy sensor, right? This is called cyclic AMP or CAMP. So this is cyclic adenosine monophosphate, right? Basically, and we eukaryotes use it as well to determine whether or not we have sufficient glucose present. Essentially, cyclic AMP levels are inversely correlated to glucose levels. So when your glucose levels are very low, your cyclic AMP levels are very high, glucose rises and cyclic AMP drops, right? So because the relationship of those two is very consistent, rather than detecting the concentration of glucose directly, you can simply detect the concentration of cyclic AMP itself, right? And use that to essentially infer how much glucose is present. And so what we're going to do with the lac operon is we're going to have an extra fine control dial, essentially, right? Which is going to be essentially boosted by the presence of cyclic AMP, right? So as cyclic AMP is high, that tells the cell that glucose is low, right? So cyclic AMP is going to increase expression from the lac operon as long as lactose is present, right? Doesn't matter if glucose levels are low and lactose is not present, you're still not going to get expression, right? So lactose is the main control of or inducer of expression from the lac operon. Cyclic AMP is the fine control. So essentially this works via another protein called catabolite activator protein, or CAP. And so what happens is, and we've got a really nice kind of look at it here, CAP is this separate protein produced from, does that say that? Oh, that's LAC-I, right? From another gene elsewhere. Don't know what that gene is called. But essentially, it binds to cyclic AMP, and when it is bound to cyclic AMP, it binds to the regulatory region and promotes RNA polymerase binding. So again, RNA polymerase cannot bind if the repressor LAC-I is present. Right? So it doesn't matter how much CAP cyclic AMP you have if LAC-I is present on the operator, right? Because RNA polymerase is not going to be there. However, if LAC-I is off the operator because lactose is present, right? Remember, oops, not that one, this one. Presence of lactose, this LAC-I is not going to bind to the operator, right? So in that case, RNA polymerase is going to start coming in here to try and bind to the promoter. And when glucose is absent or at low concentration, we're going to have this CAP-CAMP complex helping RNA polymerase to bind to the promoter. Right, so the more efficiently RNA polymerase binds to the promoter, the more transcription you're going to have. Okay, does that make sense? Anybody got any questions? Okay. Do try and keep all of this stuff within the context that I've been giving you, right? Because just as itself, it's a mishmash of words and letters and stuff, right? Okay, so initially glucose levels are low, right? Lactose is present, and so LAC-I cannot bind to the operator. Glucose is low because lac operon hasn't been expressed much. And so we've got this high efficiency polymerase binding. Now, as a consequence of lac operon being expressed, right, we're going to turn that lactose, which is present, into glucose. And galactose, don't know what galactose is involved in. I don't even know if the galactose is used in metabolism at all. I'd have to look that up. So glucose levels are going to start inching up, right? Presumably glucose is still going to be used by the cell, so it's not going to be a direct relationship because it depends on the metabolic status of the cell as well. But at some point, the production of glucose is going to start outstripping the demand for glucose from cellular respiration, depending on what that cell is doing. So glucose is going to start inching up, which means that cyclic AMP is going to start dropping, right? Remember, there's got this inverse relationship between the two. So as glucose starts rising, cyclic AMP starts dropping, and as it does, there's less of it to be bound to the catabolite activator protein. So now you could see why it's more of a dial than a switch, right? Because there are intermediates between high cyclic AMP and really low cyclic AMP. And this is going to vary too, right? Because, you know, essentially as cyclic AMP drops a little bit, RNA polymerase binding is going to be less efficient. It's still going to bind, right? But just not as regularly, perhaps, or effectively as when cyclic AMPs are really high. And so you're going to get a little bit less transcription of or expression of the lac operon, right? So as expression drops a little bit, there's going to be a little bit less lactose imported. It's going to be a little bit less lactose metabolized, and there's going to be a little bit less glucose produced. So if demand for glucose stays the same, then glucose is going to start dropping again, and cyclic AMP is going to start going up, right? And so essentially, it's like this constant, like a teeter-totter balance, right? Okay, glucose starts going up, cyclic AMP drops, right? So the boost to lac operon expression recedes, essentially, right? So glucose levels start dropping a little bit because there's less beta-galactosidase around, or even less lactose, right? Not no lactose, right? Because if that were the case, then LAC-I would bind and it would just shut it off completely, but less, right? So then, you know, glucose levels are going to start dropping and cyclic AMP start rising, and then you get more expression from lac operon, right? So as long as lactose is present, and that's the key, you could get this kind of balancing act, right? To be able to precisely match as best as possible the production of glucose, well, the expression of the lac operon to the production of glucose. And maybe at some point that lactose is all used up, right? I don't know, maybe it's like bacteria in a milk spill on the counter or something, right? And after that happens, lactose is absent, LAC-I repressor comes in and just shuts the whole deal down, right? Because there's no point fine tuning if there's nothing there to use. So that's a really good example of, you know, the big switch, right? Lactose present, on. Lactose absent, off. But when lactose is present and it's on, you then have the second question of, well, how much glucose is present? And then you can get that kind of balancing act between glucose production and lac operon expression based on cyclic AMP via this mechanism here. So this is a really cool feedback mechanism, right, in which you can negatively regulate the expression of the lac operon based on the amount of the product produced. Okay, does that make sense to everybody? Yeah, kitty cat. So what I'm going to do is I'm going to call it a day there, because it's quite tiring. Essentially, I keep hearing like the rah, rah, rah in my ear, and it's really hard to talk, kind of hear myself think in a way. So obviously after the exam today, which will be available in about 12 minutes, give or take, I would strongly suggest looking through the rest of this, right? You are going to be quizzed fairly hard on these two systems. And the lac operon is even funkier, right? So here we have this is a repressible system, right? So here tripoperon is expressed all the time, and then it's repressed in the presence of tryptophan, right? And there's also a real funky fine tuning. Actually, this is called attenuation, right? Which is probably a better word rather than fine tuning. Let's see if I spell that right. Oh yeah. Right, and so this is about secondary structures in RNA. And which structures form determines whether or not transcription continues or not. And so this is a lot funkier to kind of understand. I'd strongly suggest you look through it before we talk about it on Tuesday. And also make sure that you get the general principles and process of what's happening. Yeah, there we go, in the lac operon. Right, and so really, again, we're using these two examples because they're kind of opposites, essentially. The lac operon is inducible. The trip operon is repressible. Both have attenuation mechanisms or fine-tuning mechanisms, and the mechanism by which those occur is very different, right? Lac operon is via a second or third, maybe, protein called CAP. Trip operon is via the control of formation of different secondary structures, right? And this kind of, the trip operon goes back to transcription termination or intrinsic termination in bacteria. So you need to understand that to get how the trip operon works, or at least the attenuation of the trip operon. And I'm going to call it a day there because I'm pretty beat. My ears calling me. Yeah, I'll swallow a few of those. Like tip the pill bottle down. I'm used to having like crazy allergies because I'm surrounded by cats and a dog, and I'm allergic to both of them. But this really sucks. I don't want to have to deal with this. Anyway, and good luck in the exam. So we'll meet at 2. Let's put that up. For lab. And that is going to be, I think that's the PCR lab today. Pretty sure it is. Yeah, PCR is always fun to talk about. So we're going to meet at 2. The exam is going to be available to you. I'll just double check again that I've ticked the right boxes on it. At 12:15, and you have until midnight tonight to take it. Okay, I'll be probably be around. I'm going to go and try and take some antihistamine. Get some more coffee probably, but I'll be around for the next, I don't know, probably half hour, 40 minutes. I won't be recording, but I will be present just in case any of you have any issues with the exam. And any of you that have accommodations with DSS will have the accommodations taken care of in your exam, right? So that's, I always go through my emails and check for emails from DSS, make sure I've got everybody, so I don't forget anybody. But those are all built into that, and is actually labeled exam 3 as well. So I was pretty pleased with myself about that. Still can't believe I got that wrong last time. Anyway, that's it from me. I'm going to sign off, but I'm going to, no, 3 exam 1s would be a little bit too much, I think. I'm going to basically stop recording, but I'll leave this open. So if you do choose to take the exam when it opens up shortly, and you have any questions, I'll be around to answer them. If not, I will be around on email after the lab, but not as reliably as I will be over the next 40 minutes or so. Cool. All right.